Hello again, everybody. Right, our research on bluefin tuna is actually only just starting. So what I'm doing is I'm going to introduce a new project called WelTuna, which is the full title is Welfare Conscious Capture and Slaughter of Atlantic Bluefin Tuna in the Norwegian Rod and Line Fishery. And yeah, we just started it this year, and it's funded by the Research Council of Norway, but also by the we also receive funding from the European Partnership on Animal Health and Welfare. And partners in that includes Institute of Marine Research, NOFIMA, the Arctic University of Norway, or University of Tromsø, and Wageningen Life Science Research. So a little background. Norway actually has quite a history in terms of the bluefin tuna fishery. It had one of the largest in the 50s and 70s, 50s to the 70s. It had one of the largest tuna fisheries in the world, with nearly 470 purse seine vessels and up to 15,000 metric tons of tuna caught in a season. And the tuna we catch in Norway takes a long migration from the Mediterranean through the Atlantic, around the British Isles to Norway every summer. But in the 80s it disappeared. But early this century, early in the 21st century, we started seeing tuna coming back. And monitoring observations over time, we could see from 2015-16, we started seeing rapid increases in numbers. And these have just been increasing ever since. So in 2014, ICAT reintroduced a Norwegian quota. It was only 36 tons, and it was primarily for scientific purposes. But by 2021, that quota had been increased to 315 tons, primarily for the purse seine fishery. Also, some was set aside for bycatch in other fisheries. And what we can see, we've increased in quota over time, but in terms of the catch taken, it's actually been declining. And since 2021, the purse seine fishery has taken less than 50% of its available quota. Now, why is this? Well, we still have a limited knowledge in how to catch, limited knowledge in behaviour, and this is leading to low catch efficiency. Their fathers and grandfathers of these fishermen were the ones who used to catch the tuna. So it's a matter of re-educating the fleet. Also, limited understanding of welfare leads to poor quality and low prices. And we've also got no quality chain infrastructure in Norway, which again adds to the poor quality issues. And Izumi will be speaking more about that later. They also can't compete with the profitable herring and mackerel fisheries, which those boats are already engaged in. You can see here, at the same time as the tuna appear, we also have very active mackerel and herring fisheries, sort of towards the end of the year, and exactly when the tuna are on the Norwegian coast. And we can see the potential earnings from the quota available for all the vessels we've looked at is dominated by mackerel and herring. So they don't really dedicate capital and resources to catching tuna. But we do have an emerging rod and line fishery, which is predominantly recreational, but also we're having an increasing number of licenses. When the project initially went in, there was 2 commercial licenses. I now think it's up to 5. So there is a growing fishery. So we need to look at it in more detail. Now, what we do know about bluefin tuna is it's very susceptible to capture stress. And this can lead to poor quality, or burnt tuna, also known as yakiniku, or cooked meat is a literal translation from the Japanese. Now, we have, as you'll be aware, the members of the Catch Welfare Platform, we have a hypothesis about the linkage between welfare and quality. So capture-related stresses correlate with stress responses that increase the allostatic load in affected individuals. This initiates physiological change that reduces vitality and meat quality. And these undesirable consequences can be mitigated by improving animal welfare and handling practices. Now, the project goal is to promote ethical capture and slaughter, sustainability and catch quality in the Atlantic bluefin tuna rod and line fishery by developing animal welfare conscious capture and handling practices. We've also got a long list of other sub-objectives, which I won't go into detail now, but they will come out as I talk about the other aspects of the project. Now, our main research strategy is we have 4 main work packages: capture, stunning, welfare and tuna. Each of these will be collecting data from each and every fish. That's the aim. So we don't get many fish, so we have to get as much information from each fish as possible. We'll also have communications as always, but a very important part of the project are our stakeholders, our stakeholder group. And here we have fellow scientists from around the world. We also have the Norwegian Directorate of Fisheries in the project, the managers of the fishery. We have both Norwegian fishermen and international fishermen, as well as Norwegian fishing associations responsible in that fishery. We have experts from Japan on tuna quality, and also we have welfare NGOs. So Doug Whaley, some of you will have seen yesterday from the Eurogroup for Animals, and we also have Hugh Gollidge from the Humane Slaughter Association. Now, the first work package is capture, and here we're looking to improve efficiency, but also better understand the sustainability of the fishery. So we're monitoring bycatch, we're also monitoring fuel consumption. And this year we've used 2 boats. We've used our own research vessel, Emma Egadeus, and we've also used a commercial vessel, one of the 2 main commercial vessels, Tara. And Tara, the crew for them have the best capture rate at the moment in the fishery, so we've been targeting them. Now, in the Norwegian fishery, we're not allowed to use live bait. So what we use instead is artificial squids on spreader bars. So we have our spreader bar, we have birds, which create a lot of noise and bubbles. We have our artificial squids, and then at the very end, and normally a different colour, is the stinger, which has the hook in. And that's what we catch the fish on. And that gives you an idea of the scale of these things. So let's see some tuna catching. There you've just seen a tuna strike at the hook. That's Kino. He's the head of the capture team, and this is our tagging group. So we're basing a lot of our expertise on what these guys have learned on how to catch and tag tuna, and they're providing a lot of input on good welfare practices during capture as well. So as it says, it requires a lot of coordination within the team, particularly between the coxswain and the reelman. And we can see Kino there bringing the fish alongside the boat by hand, and then he's going to fit a hook into its jaw to bring it aboard the boat. Now this is a tagging operation. So the tuna's brought up a ramp, cover its eye to calm it down. So we've got about 300 kilos of fish there. It's tagged with a satellite tag. And then released. Now these tags stay on the fish for about a year. They're timed to release exactly a year actually after the capture date. So here they are picking up a tag which was tagged the previous year. Successfully. It's not always successful picking up these tags. So you can see Kino's very happy to got his tag back. So indeed, what does the story tell? Well, here's the results from the tagging data, and here we can see the migration. So this is the fish leaving the Norwegian coast, travelling into the mid-Atlantic. So here we are in December, January. This line actually is the ICAT delimitation between the west and eastern stocks. And it's remarkable, most of the fish are staying east of it. Now, in the spring, they've now moved into the Mediterranean for spawning, and then they quickly leave and start coming back to the Norwegian coast. So we've learned a lot about, we're learning a lot about the biology of the fish from that work. Now we move on to the stunning. This is coordinated by Ondra Grimsbø from the University of Tromsø. Here we're looking to develop humane stunning and slaughter practices. In particular, we're looking at electrical systems. There are commercial systems available. This is a Japanese system, which is a DC supply system. And we also have a Spanish system in which this ring is put around the line on long lines and sent down to electrocute the fish by touching its nose, essentially. This is a long pole with electrodes on the end for delivering the electrical field. We're also looking at developing our own system. This is going to be based more on tongs, which we put around the head of the fish when it's brought on board the boat. As a backup, we're looking at percussive stunning systems as well, very much along the lines of systems used to slaughter cattle and pigs. The first job in this particular work package is to describe in 3 dimensions the cranium and the brain of the fish. And to do this, we're going to be using an MRI scanner and CT scanner. So we're going to be taking tuna heads into our local hospital and scanning them. It's not been done before, so that's going to be interesting. And there's obviously going to be a big cleanup afterwards. Right, moving on to welfare. This is coordinated by Neil Anders, my colleague in IMR. And as Michelle pointed out, we have our pathway analysis. We've done our own. We've prioritized certain stressors. We've eliminated others because we don't think they're relevant in this particular fishery. And we're going to be monitoring environmental conditions, change in depth, temperature and salinity. We're going to be looking at behavior in detail, and I'll give you a little more on this in a while. I'm particularly looking at vitality assessments, which some of you may be familiar with from previous talks we've given. But we're also looking at the physiology, taking blood and tissue samples, taking blood and tissue temperature and pH, and also performing EEG on the fish. We're doing this with a revolutionary tagging method, which was developed by Tristan Royer, and he's collaborating with us on this. So we'll be inserting tags in the fish that are going to be killed. We'll be inserting tags to monitor heart rate during their recovery swims, before they're slaughtered, and also how quickly they die once they are bled. Now, in terms of monitoring behavior, we're trying to monitor behavior from the moment the fish are caught on the hook. So we're using these Western escape cam systems, which are placed just above the spreader bars. And so here we've just got a strike. The fish is now diving quickly. And we can get some exciting images from these, but very difficult to interpret any science from this. Our next image, we're at depth now. Now this surprised us all. What we're seeing here is tuna. It's dived down to its chosen depth. It's now swimming more or less horizontally, and it's starting to rejoin the school it left. This surprised us all. This is the fight, and the fish has actually gone back to its school. Is it seeking protection, or is it even aware that it's been hooked? These are questions hopefully we're going to answer as this research goes on. Now, this is a fish being recovered at the end of the fight. We can see it's far less active. It's quite placid on the end of the line, and we've got slow tail beats, and it's just brought up to the surface. So, as I said, these images are interesting. They're very exciting to capture. We think it's the first images of bluefin tuna on rod and line. But it's difficult to take any science from that data. So what we're looking at is also putting tags on the hooks. And these tags, the main data we're looking for is accelerometry as well as depth and temperature. Now, what does that tell us? Well, this is a sum of all of the acceleration data over time for, well, this is actually fish number 1. And we can see the strike here, and this is when the fish was brought aboard the boat. Now we can see a period here with high energy. This is in sum of gravities. So we've got high energy period initially with the strike. We've got a low energy period in the center where it appears to be quite relaxed, and then we have increased activity as it's being hauled towards the boat. Now this VEDBA, it correlates well with oxygen consumption, and that's what we're going to be using. So that's the total energy the animal is exerting into these activities. So by comparing this between fish and by identifying where the high energy periods are, we can better understand the capture process and better relate it to the physiology that we get from the fish once we get them aboard. Let's have a look in detail at some of the data. So this is the strike. Here we can see that's just the hook in the water, and we now get a dramatic increase in activity. And these are every 25th of a second. We've got data. Now, if we zoom in, this is just one axis of acceleration, and the y-axis. And what we're looking for here is particular patterns in the data. It's difficult to imagine a pattern there. Let's have a look at some other data. So we'll have a look at the calm period. So we can see this is characterized by much lower energy. We can see we're not even reaching 0.8 G. And looking at the z-axis data, we can begin to start seeing a pattern maybe. I don't know, I'm not convinced. But we're now going to look at the jackpot. This is a signal we've been looking for in the data. Hoped we'd get it. We didn't think it was realistic, but again, characterized by very low energy. And this is what we've been looking for. A nice sinusoidal pattern in the data. What does this mean, and how can we use it? Well, that's what it is. It's the tail beat. So we're picking the tail beat frequency up from the tags. That we can use to calculate speed, swimming speeds, and we can relate that to the energy the animal is putting into the fight. So, results so far. We've had 8 cruises. The guys are currently out at the moment. And 6 of those have been on our research boats, 2 have been on commercial vessels, and we've had a total of about 100 person days at sea at the moment. A little over. 8 cruises, we've caught 8 fish. So it's not a great catch rate. Of those fish, we've got 6 with video and tag, 1 with tag only, and 1 with physiology. We've not been very successful at collecting physiology because we've been focusing on the tagging. We've been prioritizing tagging for this. The physiology we were lucky to get from a recreational boat which was alongside us. They kindly let us take the fish with the permission of the Fisheries Directorate, take the fish, slaughter it and get blood samples and tissue samples from it. And it was quite a handsome catch as well at 355 kilos. We've also got 2 heads for our MRI scanning later in the year. So thank you very much for your attention. And if you've got any questions, please send them in to Michelle.
So, no, I don't see any questions at this moment. Do you have questions for yourself?
I don't have any questions for myself, no.